Good morning. I'd like to take my chance to welcome you to the summit. My name is Theo Hatzipetros. I'm the Director of Pharmacology at the Institute, and I will talk to you about some of the advancements we made last year. Where's the... Sorry. It's okay. So last year, I described to you the historical and present-day importance of this mouse to our institute. I told you that using the Black 6 SJL SOD1 G93A high copy mouse, we performed over 1,000 studies. Most of them were drug studies, but a lot of them had to do with disease biology in the mouse. I described its strengths to you, like its resemblance to human ALS, it's rapidly progressive, thus allowing us to do drug testing. But I also mentioned some of the criticisms. It hasn't translated very well into humans. I also told you that by optimizing our study design, we were able to predict clinical outcomes with 100% accuracy, albeit all of them were negative. We did that by scaling up our sample size or by balancing for gender, by controlling for gene copies, by using better statistical analysis. But we're continuously trying to learn more about the mouse. I described some of the studies we do in broadening our knowledge around the biology of this mouse, and we also share what we learn with the community. We publish often, and a lot of what we publish has been adopted by the community. And this has resulted to some positive outcomes. Specifically, we now can replicate studies of others. Others can replicate studies of us in between different labs. And finally, some of the discoveries that we made in the mouse have now made it into the clinic. Fernando touched upon those earlier. I also mentioned a few different ways that we could improve our process last year. And those were enriching our preclinical pipeline. Fernando talked about the importance of enriching our clinical pipeline, but without enriching the preclinical, you can never enrich the clinical. We also wanted to establish an early disease readout in the mouse. Needed clinical outcomes early. The same thing stands for the mouse. And finally, we needed, and we still do, an additional model, mouse or other organism, to validate what we discover in the mouse. And Fernando also touched up on some discoveries we made on that field. But this is my version of the preclinical pipeline platform. It's the third time you see this slide. This one has an emphasis on the in vivo development, which is what I do. We don't just test any compound in the mouse. The compound has to come from cell-based assays from the drug discovery group. And once we have that potential candidate, it undergoes through a series of experiments in the animals that all serve a purpose. For example, there's an experiment geared towards solubilizing in a way that can be safely administered into the mouse. And the next phase is testing it to see whether it's safe and can be tolerated by the mouse. And once we know all that, we need to know whether it engages the target that it was supposed to do. And finally, after we determine that it's good to go in a clinical, in a survival efficacy study, only then we answer the question of whether this is efficacious or not. And what I want you to take from this is two things. One, it's a term I borrowed from the computer programming people: garbage in, garbage out, quality in, quality out. If we don't have the right preclinical candidates to enter our in vivo platform, we will never be successful. The better the quality of the preclinical candidate, the more successful we will be. And the second thing that I wanted to highlight is that it takes too long to test a single molecule in the animal. Above the processes, these are the time it takes. If you add those up, it takes the better part of a year to do a single molecule. And this is not just sitting down and waiting. It involves hard work. It involves daily dosing, daily body weight measurements, daily neuroscore measurements, sometimes twice a day. So it's a lengthy, painful process. And so it's important for us to make sure that we have better candidates and find ways to make this process shorter. This way, it will allow us to test more drugs in the same time. So we were successful last year in enriching our preclinical candidate pipeline. And I'm not going to talk much about it because there's 3 posters just on that by Dr. Kyle Denton, Anna Gill, and Beth Levine. We now have the tools to produce those desirable preclinical candidates. We have human iPSC cell lines in which we screen drugs, and we have other cell-based assays that are very relevant to ALS biology. But to tell you what I meant by early disease readouts in the mouse, I have to explain more in detail the survival efficacy timeline. So we typically receive the mice at TDI from the breeder around day age 30. By age day 45, we have genotyped them, and before they turn 50 days old, we assign them to studies. At age day 50, we start dosing with the compound of interest. And then we really don't see any disease symptoms until age day 90. If we don't see any disease symptoms, we don't know if the drug is helping at all. So there's a big wait until we see some evidence of the drug working or not working. So all this phase is the asymptomatic phase of the disease. The mouse is internally sick since day 30. There's histological and biological changes happening internally, but we cannot really visualize them until they start to show signs of paralysis at age day 90. And from that point onwards, they progress relatively quickly until their end stage at around age 120. Now, if we had a way of knowing around there, or much earlier than 90, whether the changes that happen in the mouse and whether the drug alleviates those changes, we would be able to know more about the drug earlier, thus prioritizing other drugs and planning our future studies. So it's important for us it would be important to develop an early disease readout that could give us glimpses of efficacy earlier. So our answer to that is this technique called compound muscle action potential, or CMAP. For years, we struggled with ways of finding early readouts. We consider locomotion, activity measurement, but there were practical differences that did not make that possible. But we brought this technique about 9 months ago in January without having any high expectations. And so far, it seems to be working. And I will explain more about what this is. CMAP is similar to what human ALS patients undergo when they have nerve conduction studies. It is essentially a nerve conduction study. This is a motor neuron on the left, sending its axons innervating a muscle. What we do is to essentially apply a small electrical field onto the nerve and then measure electrical activity in the muscle. In the case of the mouse, we anesthetize the mouse. It takes about 15 minutes per mouse, and we stimulate the sciatic nerve, and then we measure from the tibialis anterior muscle, which is part of the calf muscle in the human. And what we typically get every time we do the test is a myograph like this one. The amplitude correlates with the strength of the nerve. The bigger the amplitude, the healthier the nerve. The smaller the amplitude, the weaken the nerve. Oops. So this technique is relatively minimally invasive, and it can be repeated time after time in the same mouse. But we wanted to make sure about that. So we designed an experiment where half of the animals received CMAP and half did not. And we measured the onset of the disease and the survival, just to make sure that it didn't impact the course of the disease in any way. And in fact, it didn't. The mice that received the CMAP 4 times every 2 weeks presented the disease at the same time as the sham animals. And also, they survived exactly the same time. I put this here so I don't forget. Everything I will show you today is based on the work of 2 people. On the left is Andy Moreno, and on the right is Josh Kidd. The next step, once we determined that CMAP is a safe technique, we wanted to design an experiment that would test what we wanted to develop to begin with, whether it can detect early deficits in the mouse. For this reason, we performed CMAP 4 times, starting at about day 50, which is the time that we typically start our drug studies, and then 2 weeks thereafter until about day 90, when they become symptomatic. And this is what we got. Even at day 50, there were significant changes from non-transgenic wild-type animals, healthy animals. This is what I mentioned here. And these changes only became worse from that point onwards. And remember, day 50 is about 40 days to 2 months before we were able to look at changes using our neuroscore system or by looking at the mice. So this bought us 2 months almost. And these changes progressively became worse. They reached a plateau at around day 90. Another thing we learned when you looked at the animals individually is that each animal was different, kind of like humans with ALS. Their rate of progression was different, and for example, in this case, these guys here, they had identical CMAP to non-transgenic mice or normal mice, while these guys were extremely debilitated even at age day 50. And most of the guys fell somewhere in the middle. But all of them were on a downward slope from that point onwards. Another thing we learned from here is that because of this variability, we couldn't just rely on absolute CMAP values to determine whether a drug works or not, but we had to express each future value as percent of baseline, or subtract the baseline value from that. So we followed up with these guys until they presented the disease and reached their humane endpoint. And then we plotted the original CMAP value against the disease onset and survival. And we found out that it correlated very well, meaning that the amount of CMAP at age day 50, which is very young, could predict when they would present the disease and when they would die. This guy here, I cannot see from here, but it had a CMAP value of around 20, and it presented the disease at age day 90 or so. While this guy with a much higher CMAP value at age day 50 presented the disease much later at age day 120. Similarly, these two guys died at much different ages, 110 versus 140. So it was very predictive, and this was very statistically significant. So far I told you that CMAP could be safely administered in mice. I also told you that it could predict onset and survival, and it can detect deficits very early, almost a month, maybe more than a month before we were able to detect. But the real question is whether we could use CMAP to determine drug efficacy. That would help us in testing our drugs much faster. To determine that, we designed a study where we used a compound that came through our internal screening program, specifically Kyle Denton's assay, which was a phenotypic cell-based drug screening assay. And this serves a dual purpose, one in determining whether that screening assay is predictive, and two, whether CMAP is predictive. So there was value in using this specific compound. I will tell you up front that currently we do not consider this compound to be a clinical candidate. It has issues related to solubility and toxicity that remain to be resolved. But nonetheless, it was a good positive control to use in this case. The experiment we did was to perform a baseline CMAP, like I described earlier, and then start dosing the drug at 3 different doses, 2, 6, and 12 milligrams per kilogram daily, and then do another CMAP measurement just 2 weeks after starting the drug treatment. This graph presents CMAP values minus baseline values. So you would expect CMAP to decrease as the mouse ages and the disease progresses. And this is exactly what we got in the untreated group. The first group here where it says 0, it means it received 0 drug. And all of these mice had decreased CMAP 2 weeks after the beginning of the experiment. In contrast, in the drug-treated groups, most of the mice did not change from baseline. Some even improved, And only a few had CMAP levels similar to the untreated mice. And this represents 3 different groups at 3 different dosing levels, 2, 6, and 12. When you combine these groups together, they're significantly improved compared to the untreated group. We followed the progress of these mice through their lives or until they reach disease onset. This is the graph similar to the one I showed you before. These are just the untreated mice. So this is the CMAP value at age day 50, and this is the time on the y-axis that they presented the disease. This mouse here had a CMAP of 20 or 30, and it presented the disease at around age day 80. You would have expected a drug that helps to delay the time that the disease appears. For example, this guy from age 80, there would be an upward shift to day 95. This is what our expectation and hypothesis was, that we would see an upward shift in that line. And this is what we saw. We did, in fact, see that most of the treated animals lived longer, or I should say, they expressed the disease later than we would have predicted. It was more obvious for the mice that had low CMAP values, or the ones that were the most debilitated. So these guys here with CMAP values lower than 40, they presented the disease later than what we would have expected when treated with a drug. A lot of the other mice did too, but some of the mice presented the disease earlier. I don't know why. But overall, our hypothesis was proved right. And the next question would be whether this compound increases survival in a survival efficacy study as the CMAP would have predicted. For this reason, we had to design a bigger study with more mice, 64 mice, gender balanced, controlling for genotype. And we dosed it at 6 mg/kg, which is the middle dose from the graph I showed you earlier, daily via oral gavage. And lo and behold, we got significant extensions or significant delays in the onset of the disease in both genders and overall, about 16 days. There was also a very significant extension in survival of about the same duration, about 16.5 days in both genders. And trust me, when I say it's significant, it's very significant. I've seen about 100 of these curves, and this is probably, if not the best, one of the 2 best I've seen. We rarely see extensions this great. Sometimes we might see a delay in onset, but then they die faster once they present the disease, and there's a net 0 value in survival. But in this case, they presented the disease later, and they died much later. And to make it even better, we repeated this, and we got the same result. So you might be wondering what next for this compound. I already told you that we do not consider it to be a clinical candidate, but through structure-activity relationship analysis, we hope to tweak its molecular structure so as to turn it into a molecule that retains its efficacy, and at the same time, we eliminate all the undesired properties. And how about CMAP? I think CMAP has proved to be a valuable tool in our arsenal, and we plan to use it going forward in our drug studies. We don't know yet how exactly to utilize it. It's a relatively new technique. Maybe we'll make specific CMAP experiments. Maybe it would be part of a survival efficacy study. But nonetheless, we plan to use it. Or it might have another function, not in drug screening, but in discovering phenotypes or changes in other mice models that do not present with a phenotype. Fernando mentioned a few earlier that we brought in the house, but we didn't discover a phenotype or a strong phenotype. One of them was the TDP43 mouse model a few years back. We discovered post-mortem that these mice had axonal deficits. They were motor neuron loss, but they died of other causes before we were able to see those. Had we had the CMAP then, we could have been able to identify those changes earlier. Another example would be the profilin mice. They look like a sod mouse, but much earlier. They're about 200 days old when they present with the disease. Kelly Mueller is working on this right now. She might be able to discover CMAP changes at 150 days. It might not seem like a lot to you, but it's going to gain us 2 months. So to conclude, I believe that I showed enough evidence to convince you that we achieved the objectives that we set for this year in enriching our preclinical pipeline, in finding early readouts, and Fernando showed evidence that we have a lot of work done on new animal models. The cell-based drug screening assay program is predictive of compound efficacy. CMAP is also predictive of compound efficacy. The TDI 1762 compound validated our entire pharmacology program since we identified it in the drug screening assay, in the CMAP experiment, and finally we took it into survival studies. And finally, we hope that through the use of SAR and vigorous in vitro and in vivo testing, we're able to make 1762 into a real clinical candidate. I'd like to acknowledge the work of many people. I already acknowledged Andy and Josh, but Val Tassinari does all the genotyping for us. Matt Ferrola and Carlos Maya take care of our animal facility. Ken Thompson, he takes care of everything. Al Gill is a great mentor for all of us, and he helps a lot with the analysis of data. Kelly Mueller is in charge of the new animal models. And finally, Kyle Denton and Matt Belukashev from the translational team, who are responsible for feeding us the compounds to test. I'd like to thank you, and I'll take any questions.